Good afternoon, everyone. My name is Suphavilai from Genome Institute of Singapore. Thanks for having me here. So today I would like to present Melias, Microbial Genome Language Model for Identification of Plasmid Sequences. We know that AMR or antimicrobial resistance is very important, right? It's a critical problem in public health. Bacteria can develop several kind of mechanisms to fight against the drug. Right? And when the bacteria turn to be resistant, then it's very hard for us to treat the infection that occur in our bodies. So usually the AMR gene is in plasmid, right? Many kind of AMR gene is in plasmid, and when the bacterial cell divide, then we increase the number of resistant bacteria. But it's not only through the cell division, we also have the mechanism called horizontal gene transfer, where the plasmid can jump between the cell, or you can even jump to the cell of different bacteria species. However, typical outbreak investigation that we do usually focuses on chromosome. So we're going to miss the plasmid-mediated outbreak, so-called, in other words, we will let the plasmid that contain AMR gene jumps around without detecting. Right. So to detect the plasmid is very important. There are quite a few number of plasmid identification methods, starting from alignment-based method that try to use the alignment to detect the known marker genes for the plasmid. Moving on to conventional machine learning methods. From the sequence, we build a set of features and then build a machine learning methods, classification model to predict whether a given contig is a plasmid or chromosome. Then we move on to deep plasmid, for example, that learn directly from the sequence itself without having to build the features. But recently, there are quite a few number of new architecture that are coming up, right? Those from language models. So we have the idea to propose this Melias framework, starting from a standard bacterial genome assembly pipeline to get the high quality contigs. Then we put this contig through the foundation models to get the embeddings. Right? Then we train a new classification model to do the specific tasks, which is we get the input as embedding and predict whether the given contig is plasmid or chromosome. But not only that, we also try to use the embedding to do the clustering. Right? We can group the embedding similar plasmid together. The cluster of similar plasmid actually can help us to flag plasmid-mediated outbreak. So what the data we have? We have collect the data of bacterial genome from RefSeq, plasmid from PlasmidDB. We train the model on the public data and test it on the public data and our in-house data, which consists of bacteria isolate that we get from both patients and environment. For the second step foundation model, we use two different foundation models here, the DNA-BERT2 and GenSLM. DNA-BERT is based on BERT architecture with more than 100 million parameters trained on human and many species. GenSLM is based on a newer architecture, GPT, having more than 250 million parameters trained on gene-level prokaryote data and SARS-CoV-2 genomes. So we want to see if we can just use this foundation model as it is to create embeddings and use this embedding in our new classification model. So in this case, we try to keep our classification model as simple as possible, 3 layers, like simple deep neural network using a binary cross entropy loss function. So let's see the performance, right? Compare the two methods that we proposed with the 3 existing methods shown in the gray bar. We found that DNA-BERT have the highest recall, while the deep plasmid that use the deep learning model trained specifically to detect the plasmid have the highest precision and F1 score. So we think that in terms of foundation model, we sometimes not use it as it is. We use it, we fine tune it. So in this case, we know the DNA-BERT was trained on human and many other species. So we took the model, fine-tuned the last few layers with the bacteria genomes. Then we found that we can actually improve the performance. You can see that the recall improved, and now we have F1 score the highest. So this is our test on public data that we have. So moving on to our in-house data. We have three cohorts of in-house data that focus on three important AMR gene type, right? We have OXA, NDM, and KPC2. So in this plot, because for in-house data, we don't have a very good ground truth information, so we want to compare the prediction between different methods instead. So this one, from the heat map, the green color represents plasmid prediction, gray is chromosome prediction, and each column represents one contig that we make a prediction. So from this, you can see that plasmid finder, which is the only alignment-based method, tend to identify the least number of plasmid, while the other four, which is the machine learning-based method, can identify more plasmid from the set of contig. And this result actually consistent with the publicly available data. The performance there, we also found that plasmid finder tend to have a lower recall. So what else we can do with this, right? After we make a prediction, okay, this contig is a plasmid. So we can go dig into the embedding of that particular contig. Here in the plot on the right-hand side, we plot t-SNE. One dot is one contig. Right? We plot the embedding together, and we can see that for the dot with the triangle shape is the chromosome, and with the circle shape is the plasmid. Different color of the circle dot show us that this plasmid contain different AMR gene type. So from here, we can actually see a few small cluster. So let's look at this particular cluster of the light blue color. Interestingly, after the manual investigation, we found that actually all of these dots have the same, look very similar. They are the plasmid size range around 71 to 84 kilobases. They share exactly the same base sequence of 71 kb. Two interesting points here that we can note. Right? The first thing is, yes, all of these contain the bacteria, but the isolate actually from different bacteria species. So if we don't see the plasmid, we only try to do the outbreak investigation based on the chromosome, we're going to miss this like plasmid that jumping around and spread the AMR. Secondly, you can see that we actually have two clusters of the light blue. This is something that is a bias inherent from the embedding itself, right? It's actually, although we have exactly the same sequence, when we put it into the foundation model, they actually try to cluster the negative strand and positive strand separately. So this is the last result that we try to understand why the model makes such a prediction, right? Given the contig, why the model predict the plasmid. Let me give some analogy to this. For example, in the language model, if I tell the model that today I am happy, and the model have to predict whether I have a good mood or bad mood. For sure, the model gonna say that I have a good mood. And the model can kind of emphasize that, oh, because I said the word happy is very important. So we use that analogy to try to identify which particular region inside the contig that play the important role for the plasmid prediction. And if you can see here, the height of the bar represent that importantness to predict as a plasmid. And we can see the similar pattern that return from both foundation models. It could be slightly different, but you can see that might be a bit small here, but the high bar will be in the region of recombinase and transposon coding genes. So to summarize, plasmid-mediated outbreak is very important. We have to do the analysis for the plasmid as well, not only the chromosome for the outbreak investigation, and machine learning really can help us to improve the detection sensitivity or recall. Foundation model can be used, and we can further fine-tune it to improve the performance, and the embedding can be used for clustering to flag out the plasmid-mediated outbreak. In terms of future work, we plan to explore a bit more on the explainability perspective. We will need more data and use case, so we are also open for the collaboration, as well as go beyond isolate whole genomic data to the metagenomic assembly datasets. So thank you very much. I would like to thanks to the study team, Evelyn Patipan, Hatay Radkaman, Niranjan Carey, and all members in the lab. Thank you very much.
Yeah, thank you for the presentation. So I have a question that you're supposed to apply the pipeline on the metagenomic assembly, right? So it is very likely that the metagenome assembly is very fragmented, even for plasmid, and but your training data are all on, if I understand correctly, intact plasmid. Okay, so do you try to check how fragmented can you still predict the plasmid based on the language model? Thank you.
Thank you very much. I think that's a very, very good question, and it's actually one of the direction that we want to study as well. Yes, you are right that for now it's almost intact, fragmented, and we really actually try to running the simulation to firstly run the simulation to see the boundary kind of what are the kind of minimum length that we actually can make a prediction. There are a few existing tools that they even use the length with the fragment length as a threshold, right? But we try not to use that. We try to go purely based on the content of it. So we have to kind of find a boundary at which minimum length that we have enough content to make a prediction. And I don't have an answer for that yet, but that's one of our direction to go toward the metagenomic. Thank you very much.
What kind of plasmid could you detect by Melias and Melias, but not by previous one? So what feature these plasmid have?
The question was what kind of plasmid that can be detected by our method Melias but cannot be detected by other methods. My answer would be this plasmid actually cannot be detected by plasmid finder. It could be because the plasmid finder really focus on a subset of species, and it contain only the database contain only the marker from those species, and it missed this all kind of marker. Although we find all the transposon, all the recombinase inside the plasmid, but it somehow missed it. But actually, all other machine learning model actually can detect this.